Broadcast is now starting. All attendees are in listen-only mode.
Hello, everybody. We would like to get started with this webinar at this moment. My name is Hideyuki Terashima from the University of Illinois, and I will be moderating the November edition of the Joint Centers webinar. This webinar provides an opportunity for Centers students to present their research. We will have three 15-minute presentations, each followed by a 5-minute Q&A session. So while the presenters are presenting, feel free to ask any questions using the webinar or GoToWebinar's question chat box feature. This presentation will also be recorded and will be posted on the Drinking Water Center's website at a later date. Our three presenters for today are Lee Gilmore from University of Colorado, Suyun Kum from University of Texas at Austin, and Siddharth Bhartia from RESEAU. So at this moment, I would like to change my screen control over to Lee. Feel free to begin when you're ready.
Thank you. Great, thank you. And thank you all for attending this webinar. My portion is titled Biofiltration Performance, Evaluating and Modeling the Effects of Extended Empty Bed Contact Time, Temperature, and Dissolved Organic Matter Character. The co-authors on this work are Dr. R. Scott Summers from University of Colorado. So drinking water biofilters occur when bacteria develop in filters without the presence of a disinfectant residual. The naturally occurring heterotrophic bacteria attach to medium such as sand or anthracite and oxidize the dissolved organic matter for energy supply and a carbon source. The attached biomass is comprised of active microorganisms, which are oligotrophic in nature, meaning they can survive on limited primary substrate, as well as extra polymeric substances excreted outside of the cell. And this biomass can degrade dissolved organic matter, as well as other micropollutants. Biofiltration also improves water quality and stability by lowering regrowth potential as well as lowering corrosion potential and increasing residual stability in the disinfectant system. Here in this diagram, you can see a flow biofilter column, and you can see the anthracite with the EPS and the bacteria attached around the anthracite. The following slide shows a conventional surface water treatment train for drinking water. You can see the filtration, that disinfection is normally added before, and depending on the influent water quality for the system is required to remove anywhere between 15 to 50% of the organic carbon. However, with biological filtration, you can move this disinfection point into after the filter and allow the naturally occurring bacteria to develop. And this can provide an additional 10 to 15% of organic carbon removal. So organic matter in our drinking water source can come from different avenues. One is a terrestrial source. So organic matter is derived outside of the water body and brought in. Another source is microbial, which is organic matter derived within the water body. And then wastewater effluent, organic matter, is when you have a wastewater effluent discharge upstream of your drinking water intake, and it will produce micropollutants, which are anthropogenic or natural in nature, and soluble microbial products, which are derived within the wastewater treatment process. So why do we care that these are in our water? Well, dissolved organic matter can affect water treatability. It affects your oxidant demand and your micropollutant removal, as well as water quality, DBP levels, taste, and odor, and color, and water behavior, bacterial regrowth in the distribution system, and you can lose your residual disinfectant within the distribution system, and it can lead to corrosion. Disinfectants, such as chlorine and ozone, react with pathogenic microorganisms to inactivate the microbes, and this is an intended reaction. However, whenever there's natural organic matter or inorganic compounds in the water, these react with the disinfectants to form disinfectant byproducts, which are harmful to humans, and that is an unintended reaction. So this work looks at reducing the natural organic matter in order to form less DBPs. This work also looked at modeling biodegradable dissolved organic matter removal within the biofilter. So here you see a schematic of a downflow biofilter. Empty bed contact time is the volume of the empty bed of the filter divided by the flow rate. And we used Monod equations. We combined the cell growth as well as substrate utilization to derive the change of concentration over the time it spends in the biofilter. As you remember, the substrate coming in is extremely low, so this concentration of the limiting substrate for growth will be less than the Ks with the high half velocity constant. Therefore, it is insignificant. And we can set the contaminant utilization rate constant equal to the maximum specific growth rate of the microorganisms over yield coefficient times the half velocity constant to have the simplified expression, where the change of concentration over the change of the time it spends in the biofilter is equal to the contaminant utilization rate constant times the biomass concentration within the filter times the concentration of the contaminant. We integrate this equation to see that removal is an exponential function of the following parameters. In order to adequately model biofilter removal, we need to know the biomass concentration within the filter. And with this equation, that's represented by X. So we went to the literature and we found 78 studies for non-ozonated biofilters, and we plotted normalized biomass at specific empty bed contact times to biomass at the top. So empty bed contact time is on the X and the normalized is on the Y. If the biomass was remained constant throughout the filter, the value would be 1. However, this graph shows that the biomass decreases with depth. At about 5 to 6 minutes empty bed contact time, biomass is already 50% of that at the top. We also looked at ozonated studies, as shown by the dark squares. And you can see that, again, biomass is decreasing with depth, with 50% at 3 minutes empty bed contact time. So therefore, the biomass decreases at a steeper rate than for the ozonated studies than the non-ozonated studies. So with this, we could dictate that biomass at a specific empty bed contact time, if we know biomass at the top, times the two A and B are constants derived from the literature, and your empty bed contact time. We integrate this equation to derive total biomass, so you can know the total biomass throughout your filter by knowing biomass at the top and the different A and B constants. So this study, we had the following bench scale matrix. For our influent water, we used a terrestrial source as well as microbial and the wastewater treatment plant effluent. And in addition, we used baseline concentrations of 3 milligrams per liter for each of the waters. For empty bed contact times, we looked at 5, 15, and 30 minutes. For the biomass study, we looked at the dissolved organic matter source as well as other pretreatment options. And we looked at 3 different temperatures, 7 degrees Celsius, 22 degrees Celsius, and 28 degrees Celsius, in order to encompass the range that a water treatment plant could see during a year of variability. And we assessed dissolved organic carbon, absorbance, nutrients, alkalinity, pH, and activity. This was the setup of our bench scale schematic. We had 3 columns in series. You can see the empty bed contact times are 5, 15, and 30. We recirculated the water for a period of 4 days with valve 1 open, and then we changed out the feed water and single pass for about 4 hours and then sampled. In order to control temperature, we had a cold room set for 6 degrees Celsius. The lab temperature was at 22 degrees Celsius, and that cold room could also be converted to a hot room set for 28 degrees Celsius. So this is how we were able to control temperature throughout the experiments. For a terrestrial source, we used Barker Reservoir, as seen in the photo on the right. You can see ski slopes in the background of that photo. So this reservoir receives snow melt infiltration yearly. It had a SUVA of around 2.92 liters per milligram per meter. And this water is one of the source waters for the city of Boulder, Colorado drinking water treatment plant. The microbial source we used for this study was Wonderland Lake, as seen in the photo on the right. It is a purely aesthetic lake, meaning there's no infiltration from runoff. The SUVA of this lake was 1.6. And then lastly, our effluent organic matter source, we went to the Boulder wastewater treatment facility and collected the effluent. You can see in the top photo, the blue circle, that's where the effluent discharges into the creek. Therefore, we set up a recirculating PVC pipe in order to recirculate anthracite to the specific water matrix for 2 weeks. And then we were able to bring that back to the lab as well as water to run the test. And this water had a SUVA of 2.1 liters per milligram per meter. So in order to get into some results, we wanted to look at activity. We used the LumenUltra test kit, where ATP reacts with luciferase enzyme found from the firefly to produce light. And that light can be correlated to nanograms of ATP per centimeter of media. So for each of the top of the columns, we are able to take a media sample and measure activity. In this chart, you're seeing activity in nanograms of ATP per centimeter cubed media, and then each of the sample points, top, 5-minute empty bed contact time, and 15. This green bar shows the Boulder wastewater effluent water. So the top had around 900 nanograms, and then it decreased with bed depth, which is what we saw in the literature. Next, we wanted to add on the Wonderland Lake activity. We see that it's less than 600 nanograms of ATP per milliliter of media at the top, and again, it decreases with depth. And then we added Barker Reservoir, and it was a little less than 500 nanograms of ATP per milliliter of media at the top, and again, decreases with depth. So this is the same trend that we saw in the literature studies. It's interesting to note that the Boulder wastewater effluent had the highest activity. So we'll have to see how much removal we get from Boulder. Next, we wanted to look at dissolved organic carbon removal results. This was the terrestrial source, Barker Reservoir. You see empty bed contact time on the X in minutes and DOC removal percent on Y. The blue bars represent the cold 6 degrees Celsius temperature. 22 degrees Celsius is shown in purple, and 28 degrees Celsius is shown in red. So we can see that with increasing temperature, we're increasing our DOC removal at each empty bed contact time. In addition, with increasing empty bed contact time, as the EBCT goes from 5 to 15 to 30, we're seeing an increase in DOC removal as well. So next, we looked at coagulated water at 22 degrees Celsius, shown in the green, as well as the diluted, the blue bar at 22 degrees Celsius. And this was the control, baseline control, where we did 3 milligrams per liter of concentration. So we saw 12% removal for Barker. We'll want to compare this to the ones going forward. So we can see, we've set this to modeling data, and we have total activity, which we use from those equations I showed earlier, compared to DOC removal. We plotted each of the different scenarios. And then we plotted our best fit K double prime to be 1.8 with a BDOC of 20%. Next, we looked at our microbial results. Wonderland Lake, we see again empty bed contact time plotted against DOC removal. And we see the same trend with increasing temperature leads to higher DOC removal percent, as well as increasing empty bed contact time. When we looked at our control, where we set the TOC influent to 3 milligrams per liter, we saw 12% removal, which was the same as Barker. We applied our modeling techniques, and we found a best fit of K double prime of 1.3 times 10 to the negative 4 with a BDOC of 20%. Lastly, we looked at our wastewater treatment fluent effluent. We see again the same trends with higher temperature leads to higher removal, and higher empty bed contact time leads to higher removal. For the baseline concentration, we saw 12% removal, so that it was the same throughout each water source. And then we looked at total activity versus DOC removal for each of the scenarios, and found a best fit of K double prime of 1.5 times 10 to the negative 4 but yet the BDOC of this water was only 14%, not 20 like the other waters. So some takeaways from this study, we saw that higher temperature led to increased DOC removal, as well as longer empty bed contact time, increased DOC removal. We saw that SUBA in the range of 1.6 to 2.9 did not seem to impact DOC removal, as our control saw the same 12% removal in each source water. And the kinetics were similar in all three source waters, despite equal DOM concentration and different DOM character. In addition, we saw that biofilters are robust in removing DOC from a range of DOM sources, which is beneficial for drinking water treatment plants, which pull from a range of sources. And with that, I'd like to acknowledge the U.S. EPA STAR De-Risk Center, as well as the STAR Fellowship, Lumen Ultra, the City of Boulder, City of Longmont, as well as Scott Summers Lab Group. And with that, I think we have 5 minutes for questioning before we switch to the next presenter.
Thank you, Lee. That was a very interesting presentation. I had personally a question. Did you ever consider seasonal changes? I know you tested with different temperature, but I was wondering if you tested the water source like from one specific season as opposed to a different season. I don't know if you consider that to be some sort of effect on your the treatment.
Right. We did that at in a different study at the pilot scale. So not necessarily in this study because we sampled the different sources just one time and then ran all of the tests on that one large barrel of water. But we have looked at different seasonal variability at the pilot scale, but that's a different study.
Oh, very nice. Let's see. And it looks like we don't have any other questions at the moment, but
okay.
For the audience, if you have any other questions like that might come up later on, feel free to ask. At the very end, we can go over overall Q&A session as well. So at this moment, Lee, if you don't mind changing screen controls to Cyan.
Okay. Everybody can see my screen?
I personally cannot see your screen right now. I don't know if your show screen option is grayed out right now or... Why don't you change a presenter back to me, and I can try setting it to you personally. Perfect, we can see your screen now.
You can see my presentation fine, right?
Yep. Once you go into presentation mode, it should be perfect.
Mm-hmm. Hello, everyone. My name is Soyun Kim, and I'm at the University of Texas at Austin. Today's my presentation topic is about coupled ED-NF system to remove disinfection byproduct precursors, mainly about natural organic matter. And this is the part of a WINS project. So natural water contains the natural organic matter, and natural organic matter itself is not that problematic. But in the water treatment plant, when natural organic matter meets a disinfectant such as a chloride, it can form the disinfection byproduct at the distribution stage and can supply to the tap water. So we can remove the natural organic matter, or we can change the disinfectant to prevent the DBPS, and we also can remove the DBPS. But in my research, I focus on natural organic matter removal, and to remove the natural organic matter, we can use a coagulation fluctuation, ultrafiltration, nano-filtration, and reverse osmosis. Among these processes, Nano-filtration and reverse osmosis shows the effectiveness to remove the natural organic matter. So nano-filtration and reverse osmosis, pore size, NF is 1 to 10 nanometer, and pressure for the membrane system is 50 to 150 psi. And advantage of nano-filtration is can remove dissolved organic matter. But the disadvantage is fouling increases when NAM coexists to hardness. Reverse osmosis pore size is smaller than nanofiltration, so it requires much higher pressure. The advantage is it can remove almost everything in the water, but disadvantage is also it removes almost everything water. So at the end of the process, we need to put minerals, and also it has a fouling problem. And here's one more technology, the electrodialysis, which size is really small, but it doesn't require the pressure. So electrodialysis uses electric current to separate the ion in the water. So the advantage of ED is it can remove both the divalent and monovalent ion. But disadvantage is it's not effective to remove the natural organic matter. So in our research, using the advantage of NF RO-ED, we suggest coupled ED NF RO to remove NF not effectively. So here's more detail about coupled ED NF RO system. So first, we put the natural water to electrodialysis, and then we can get the ED dilute with really low ion concentration, but unchanged NAM concentration. And here the dilute means the clean water, to produce the water from the ED. And then we put the ED dilute to nanofiltration or reverse osmosis, then we can get large amount of NF-RO permeate. Permeate means the produce the water from the NF-RO, and the permeate has a really low ion concentration and also really low NAM concentration. And we can also get the NF-RO concentrate, really small amount of concentrate, and with the high ion and NAM concentration. And at the end of the ED-NF system, we mix the small amount of ED concentrate. Concentrate has a high ion concentration, but we unchanged the NAM concentration from the original water. And we mix this water with the permeate from the NF and RO, then we can get the produced water with the 70% of NAM and 14% of ion reduction water. So let's talk about the electrodialysis first. Electrodialysis consists of a cathode and anode. So in between the cathode and anode, we install the cation exchange membrane and also anion exchange membrane. And we supply the feed water in between the cation exchange membrane and anion exchange membrane. And we apply the electric current to separate the ion in the water. So in the stack, the cation cannot pass through the anion exchange membrane, but it can pass through the cation exchange membrane, and vice versa for the anion. So at the end of the stack, we can collect the almost ion-free water, which is called the dilute, and also on the other stream, we can collect ion-rich water, which is called the concentrate. And here, this is the basic the theory about the electrodialysis, and we add the NAM in the electrodialysis as well, but because of the diffusivity of the NAM, it doesn't separate in the electrodialysis. So both the dilute and concentrate has a pretty much a similar concentration of NAM, like feed water. So ED needs for the coupled ED ADAP or RO system is a good separation of ion like general ED system, but it requires a minimum non-removal and minimum non-absorption on the membrane surface. So it requires understanding of non-properties and also ion exchange membrane properties. So I use a battery recycled ED system. So I recirculate the dilute and also circulate the concentrate, and I have electrode rinse, which keep maintain the same pH at the cathode and anode. So at the beginning of the experiment, both the dilute and concentrate start from the same feed concentration, but at the end of the experiment, dilute has a really low ion concentration, but the concentrate has really high ion concentration. I control the flow rate and also measure the flow rate and measure the pressure at each stream, and also I measure the pH, conductivity, and temperature at each stream, and sampling the water every 10% conductivity removal. Here's the experimental matrix water composition. I use two different types of the base solution, one called the soft water and the other one called the hard water. So soft water and hard water both contain the sodium and chloride, but hard water also contains some calcium and magnesium. Also, I run the system with no NUM condition at the Swanee River NUM, which has a SUBA number 3 to 4.5, and also I add the leonardite humic acid, which has a SUBA number 9 to 11, indicates that more hydrophobic than Swanee River NUM. Here's the ion exchange membrane information that I use for the ED system. One is the PCSK and PCSA membrane. I always use the pair, the cation exchange and anion exchange membrane. And the other pair is the CMX AMX membrane. And third one is the CR67 and AR204. The ion separation characteristic affected by ion exchange capacity of each ion exchange membrane, and the NUM separation mainly affected by membrane material. Here's the full experimental apparatus photo, and here's the result of the soft water. I'm going to talk about the PCSK and PCSA membrane with the 5 milligram per liter DOC of the Swanee River case first. So left to y-axis is the concentration ratio, and the right y-axis is the UV absorption ratio. And the x-axis is specific energy, which means amount of the energy consumed by the membrane process to produce given volume of water. So when we use more energy, we can get the lower concentration of permeate, which is the open diamond line. So you can see the PCSK and PCS membrane that separate the ion really well. This one is the sodium because it's soft water. And you can see the UV absorption is nearly 1, which indicates that ED doesn't separate the NUM in the water. So let's see the old data, no NUM condition, Swanee River and leonardite humic acid. The ion separation is pretty good, and also UV result is nearly 1, so not that much in NUM separation in the PCSK and PCSA membrane. Here's all 3 membrane pairs experimental result. So PCSK and CMX membrane pairs pretty much similar result, but on the other hand, the CR67 and AR204, you can see the absorption ratio is decreased at the higher specific energy. So to confirm that this result, we check the TOC result. So here's the NUM recovery at the end of the experiment. CMX AMX NUM recovery is almost 1. It's like 28% to 27% at Swanee River NUM and leonardite humic acid. The second one is the PCSK and PCSA but CR67 and AR204, like the UV results, it shows really poor NOM recovery. So we excluded this membrane pair for hard water experiment. You can actually see the NOM composition on the membrane. At the right side membrane is the anion exchange membrane, and since the NOM has a minus charge in the water, it observes on the anion exchange membrane. Here is a hard water without NOM condition. So you can see the excellent removal of calcium and magnesium as well as the sodium at both membrane. And when we see the NOM recovery data, like the previous soft water case, the CMX, the AMX shows the better NOM recovery. So I can say that CMX and AMX is appropriate to couple the ED-NF RO system. Not only CMX, AMX membrane, the type like CMX and AMX membrane is good for the ED and NF system. The next thing is the nano-filtration part. Here's the nano-filtration stack. I use a flashing membrane and also the cross-flow nano-filtration. So I supply the feed water and then supply the pressure. Then I can, the water pass through the membrane and I can collect the permeate like this way and then concentrate at the bottom. And the NF needs for the coupled ED-NF system, I need to reduce the fouling by NOM and inorganics. So requires understanding of water chemistry of feed water and NOM properties, NF membrane properties. So I ran the two different type of NF experiment. Here is the first one is a general NF experiment, and the other one is NF fouling experiment. So here's a schematic diagram of general NF experiment. I collect permeate to the mass and measure the mass of the permeate and log the data through the computer. And also I measure the before and after nano-filtration pressure and measure the pH, conductivity, temperature at the feed solution, and measure the flow rate at the system. The NF fouling is almost everything was the same, but I recirculate the permeate. So recirculate the feed water for 80 hours. So here's a more detailed experimental condition. I use the membrane NF270, which is commercially used for organic removal, and applied pressure was 70 psi, pH was in between 6.5 to 8, and flow rate was 1200 to 1300 milliliter per minute, and operation hour for general experiment was 9 hours, and fouling experiment was 80 hours. And feed characteristic, I used 5 milligram per liter DOC Swan River NOM, and this TDS, I used 500 milligram per liter, which indicate without ED condition, and 185 milligram per liter, which indicate with ED condition. So for general experiment, without ED condition and with ED condition shows a pretty much similar result. So both of them, I can achieve the more than 90% removal of the NOM in the system. For the fouling experiment, this is the permeate flux change along the time. So the y-axis is a normalized flux, and it decreased when I run the system for a long time. So when I compare the both with ED condition and without ED condition, with ED condition is a little bit above the without ED condition. So it indicates that there is a less fouling at the with ED condition. Here is a conclusion. The ED removes a little NOM while achieving high removals of inorganic cations and anion, and the CMX-AMX membrane pair shows the lowest NOM absorption and effective cation separation. And NF removes over 90% of NOM, and ED pretreats delay the NF fouling. So we can conclude that coupled ED and NF is the promising technology for non-removal from natural water, and we can remove 70% of low ion removal from the original water. And this coupled ED-NF is applicable at any size of plant, but recommended for the small size water treatment. Thank you for the listening. Any question?
Thank you, Soyeon. It looks like we don't have any questions at the moment, so I guess we'll be moving on to our next presenter.
All right. Thank you so much.
Hi everyone, I'm Siddharth and I'm doing my master's at University of British Columbia, and my project is part of Reservoir. So basically we are investigating the water quality changes for vacuum UV advanced oxidation treatment in drinking water, focusing on biological stability. So to give a brief background on UV-based AOPs, UV254 along with H2O2 is extensively used for micropollutant degradation. It's highly popular and many industries and municipalities use it for the treatment. But whenever we talk about small systems and small communities, there are a few issues and challenges associated with this because it uses a chemical. Now, most of these small communities are remotely located. They are not well equipped in capacity, and the transportation is always a challenge. It adds to the cost, and then there are like weather challenges, like wintertime, it's tough. Hence, it becomes important and interesting to study a chemical-free advanced oxidation process, like just using UV, no chemical oxidant. Now, this can be facilitated using vacuum UV. Vacuum UV corresponds to 100 to 200 nanometer range in the electromagnetic spectrum. As we can see, it is the lowest wavelength region in the UV. So because of the lower wavelength, they have high energy, and we can use this approach to split the water molecules used, in this case, 185 nanometer photons to generate OH radical, and thereby getting advanced oxidation process. Now, vacuum UV for degradation of micropollutant has been extensively studied by a research group with many model pollutants, including pesticides, pharmaceutical, and cyanotoxins. Now, in this slide, we see two such examples. The first graph shows degradation of 2,4-D. 2,4-D is a chemical found in herbicides, and in the graph, we can see 2,4-D concentration in milligrams per liter on the y-axis. X-axis has the radiation time for vacuum UV. And here experiments were carried out using different surface water from British Columbia. And we see steady degradation of 2,4-D with the vacuum UV application for all different surface water. The second graph is MCLR, or microcystin LR. Now, MCLR is an algal toxin. It's highly regulated in drinking water. And here we see 254 in combination with 185, and we see degradation of MCLR at lower UV dose when 185 is combined with 254. Now, vacuum UV offers some advantages in advanced oxidation process. Firstly, it's very similar to UV disinfection, like you need some little modification with the lamp, and the electrical supply, the input, is more or less similar to UV disinfection. It's very simple to operate because it doesn't require any chemical, hence less job in terms of operators. And apart from degradation of micropollutant, it also caters to the disinfection needs in a drinking water system. Hence, oxidation and disinfection takes place together. But whenever we talk about advanced oxidation process, be it ozonation, UV peroxide, there's always a challenge of biological stability, and so be with vacuum UV, because the OH radical that's generated is non-selective, and apart from the target micropollutant, it also reacts with natural organic matter or NOM in the water. Now, we can get complete degradation of NOM, but at a higher UV dose, which would lead to an increase in the cost. Hence, it's not possible practically. Therefore, at the applied UV dose, the partial oxidation generates smaller molecular weight fractions, which are more biodegradable, that can increase the assimilable organic carbon concentration. Now, AOC is responsible for the bacterial regrowth in the distribution system. The bacteria present in water can use AOC as a food source for the rapid growth. Hence, if we have high AOC in our water, that means it's reduced biologically, so, it's less in biostability, thereby a potential health risk. Hence, the objective of our research is to study the impact of vacuum UV process on biological stability, which are measured by AOC and BDOC. Biodegradable dissolved organic carbon is another parameter for biological stability assessment. And as the term suggests, it's the biodegradable component of DOC in the water. Further, we also study impacts on DBP precursors, which is analyzed by chlorine demand and DBP formation potential. Further, we evaluate biological activated carbon process downstream vacuum UV, aiming improvement in biological stability by reducing the AOC and BDOC in the water. Now, for our vacuum UV experiments, we used two setups. The first one was a collimated beam setup, as we can see in the picture. This is used for kinetic studies, and it uses a small volume of water in a beaker, as we can see, and UV radiation hits from the bottom, and there is a mixer that keeps the solution homogeneous. Also, we use a flow-through bench scale setup to evaluate the water quality conditions at different operating conditions, and the AOC concentration is determined using flow cytometry analysis. Flow cytometry is one unique way of measuring AOC. It uses bacterial cell count technique, and the AOC concentration is determined using calibration plot, or we call it as the yield coefficient. Here, the cell count is analyzed by staining the DNA of the cell with an organic dye, and thereby the flow cytometry counts the cells in the water with fluorescent light scattering method. Here we see the schematic for the bench scale flow-through reactor. The solid lines we see are the water connections, while the dotted lines are the electrical connections. We focus on the solid lines, or the water connection. So water flows into the reactor all the way from 1 to 3 and enters the photoreactor at 4. We have the lamp, which is in blue color, the VUV lamp, and it's protected by a cord sleeve, and water flows on the surface of the sleeve being radiated by UV, and then it's collected at number 6, at point 6. On the right, we also see the actual setup, how it looks in the lab. And yeah, this was used for evaluating the AOCs and the DBPs by changing the flow rate. Thereby, we get changes in the residence time of the UV radiation. Now, for our experiments, we used real surface water. We used 3 different surface water: Middle River, Vananda, and Cedar Creek. These are source water for various small communities located in British Columbia. Now, Middle River and Cedar Creek are in fact the First Nation Aboriginal communities here, and The TOC for Middle River and Van Anda were in range of between 5 and 6, while Cedar Creek was lower, almost half of them, like at 2.7. Rest of the properties, pH, UVT, were more or less the same. Now, this graph shows the AOC formation kinetics using the collimated beam apparatus. On the y-axis, we have the AOC. On the x-axis, we have the UV dose ranging between 100 and 1000. Commercially applied UV dose ranges between 500 and 1000 millijoules per centimeter square, which is sufficient for most of the micropollutant degradation. Hence, we restricted our kinetic study range between 100 and 1000. Now we see a steady increase in the AOC profile for all different surface water used in the study. To further understand and to simplify the data, we normalize the AOC concentrations at respective UV dose with the initial AOC concentration of the water. So this is the normalized graph, and here we see a linear fit of all these data. In fact, if we see closely, the AOC increases is between 1.5 and 2.5 for these three water, and the linear fit of these data points suggests that the AOC formation follows a zero order kinetic with respect to the UV fluence. Also, it was found that the slope of these linear fit were in proportion with the DOC concentration of the water, thereby suggesting that the rate constant of this reaction is dependent on the DOC or the NOM of the water. Now this slide shows the AOC and BDOC data using the flow-through reactor. On the first graph, we have the AOC concentration in parts per billion or micrograms per liter, while on the x-axis of both the graph, we have the residence time ranging 5, 10, 20, and 40 seconds. And the corresponding UV dose with respect to these residence time were in range of 400 to 1800 millijoules per centimeter square. And now here we can see like how quick vacuum UV process is. Like for example, 40 seconds gives us a UV dose of 1800 millijoules per centimeter square. Also, we see a steady increase in AOC with the UV dose, and similar trend is seen for the BDOC profile as well. And the extent of growth in BDOC is higher in Middle River water as compared to Cedar Creek, because Middle River has a higher NOM, or which was represented by the higher DOC in the water. And these data confirm the impacts of vacuum UV on biological stability because of the smaller molecular weight fractions formed. Now, coming to DBP precursors and impacts on them, we analyzed chlorine demand and DHM formation potential. So chlorine demand is the amount of chlorine required in disinfection process, maintaining a residue amount. And we performed 24 hours chlorine demand, followed by DHM formation potential analysis at uniform formation condition. And here also, similar to AOC and BDOC profile, we see a steady increase in chlorine demand and DHM results, suggesting that the smaller molecular weight fractions, apart from impacting the biological stability, also is a precursor to DBP byproducts. And for both these data, we see higher increase in Middle River as compared to Cedar Creek, again because of higher NOM present in Middle River water. Now, as a mitigating step and to enhance the water quality, we coupled biological activated carbon treatment post VUV, and as we can see in the picture, lab scale BAC columns were designed and integrated after vacuum UV process. The empty bed contact time, or EBCT, was 20 minutes for all these 4 columns, And it was found from literature that 20 minutes of empty wet contact time should be sufficient enough to reduce the AOC and BDOC level and improve the water quality. Now I'll just show like two results from this. So the first one is the BDOC profile, and the second one is the THM formation potential after combined vacuum UV and BAC. Now for both these graphs, the blue column represents the data points after vacuum UV process at different residence time of UV radiation, while the orange columns are for combined vacuum UV and BAC treatment. One thing to note that the orange column corresponding to this raw water is the BDOC value after only BAC treatment, like no vacuum UV. So this act as a control to see what's the change after application of just BAC. And for the BDOC data, we see significant BDOC removal after combining with BAC, and similar results were seen for THM as well. In fact, when we analyze these data, the difference of BDOC and THM removal were in proportion to the influent values through the BAC column. And that means like higher the biodegradable fraction entering the BAC column, higher is the extent of removal. So these data clearly suggest that yes, biological activated carbon treatment is efficient in improving the water quality. Thereby, vacuum UV and BAC is a feasible option to be used in water treatment. And to quickly summarize the result and what we saw in this presentation, so VUE offers a chemical-free alternative for advanced oxidation treatment process, and it's very efficient in degradation of micropollutant. We saw examples with 2,4-D and MCLR, but unlike any other AOP, it does impact surface water quality because of the partial oxidation of NOM. But this challenge can be easily mitigated using a simple technique, in this case like a BAC treatment post VUV. Hence, overall, vacuum UV is a suitable alternative for the small systems, and the unique selling point about this is that it caters to both disinfection and degradation of micropollutant in the water. Finally, to thank our funding partners, RESEAU, WaterNet, and then people at UBC, All Polytechnic, and Credo. And thanks everyone for your attention.
Well, thank you for that presentation, Sid. We'll be moving on to our Q&A session now. Sid, have you noticed any change in the UV system, the bulb lifetime at all, when you introduced the vacuum component to it? Were you able to elongate the UV bulb's life expectancy, or did it decrease at all?
Generally, like we have a fixed lifetime, like specified by the supplier for the lamps. It ranges, like it depends on the lamp. It depends, like for example, it can be 10,000 hours in some case, and some might have like 20,000 hours. But yeah, I didn't do much study on the lifetime of the lamp.
I see. We also have another question. Oh, sorry, go ahead.
Yeah, and also like we use smaller lamps for the lab scale experiments, but for like bigger lamps and might have a different lifetime, like and Trojan Technologies are the supplier for the UV lamps. So in each, the lifetime depends with cases, like depending on the lamp model and other factors.
I see. We also have another question. For the BAC, was it fresh carbon or was it acclimated?
No, it was exhausted like carbon. So we just had like a biological process, no absorption taking place. In fact, I monitored the DOC level as well, and we saw like 20% decrease in the DOC, which is accounted for just the biodegradable fractions.
Right, perfect. That's all the questions we have for you, Sid. Suyun, we actually have two questions for you if you
All right.
are still there. For question number one, for the nanofiltration and Ross osmosis, did you for the fouling issues, or they had fouling issues, but for electrodialysis, was there fouling involved in that as well?
Yeah, it depends on the ion exchange membrane. So the purpose of that I tried the different ion exchange membrane is try to find ion exchange membrane which has the least fouling problem by NOM as well.
Perfect. And how is electrodialysis efficiency affected by the initial ion concentration of the water?
It normally used for the brackish water desalination. So if the ion concentration is not like seawater, the efficiency of the ion separation is pretty good at electrodialysis.
All right, perfect. Well, at this moment, we're out of questions, so I would like to go ahead and conclude this session. I would like to thank our three presenters today and for everyone else that attended this webinar. Thank you and have a nice rest of the day.
Thank you so much.
Thank you.